I now have the privilege of introducing my friend and colleague, Dr. Amit Etkin. He's an associate professor and an associate chair in the Department of Psychiatry. An MD, PhD, you're going to see a bunch of doctors today, where he's a psychiatrist and a neuroscientist. I had the really wonderful good fortune of having Amit join the faculty right as I arrived at Stanford, and he immediately received an NIH Pioneer Award, established an amazing laboratory, really a stellar trajectory. His interest is in developing greater knowledge of human brain circuits in health and in disease states. And I also want to acknowledge that Amit helped us plan the meeting today and to recruit many of the speakers and many of you. So let me introduce Amit Etkin.
Thank you, Laura. Thank you to Michael. Thank you all for coming. This is really going to be the beginning, I think, of a very exciting weekend. I look forward to all the conversations we'll have together. But let me start for you with a $6 trillion opportunity. This is the cost associated with mental illness worldwide by 2030. It's so high because psychiatric disorders are common, chronic, and disabling. Depression, for example, tops the list in terms of burden. In fact, if you zoom in on depression, you see that even though we have multiple effective treatments for this condition, still 1 in 3 people do not respond to medications even after multiple trials. This is a big and hard to solve problem. To try to understand some of the challenges ahead, it's useful by analogy then to look at other fields and think about things outside of our own sphere. So let me tell you a story about aviation. This is the Boeing 247. Interesting airplane from various regards, but if you look at its cockpit versus that of the Boeing 787 that we fly in now, you see a vast expansion of technology, computers, GPS, and the like. I'd like to say that this is the path that we're on in psychiatry, but unfortunately, I don't think that's entirely true. I think it's actually closer to this, the Spirit of St. Louis. By all measures, an incredible airplane was able to get from New York to Paris. But the difference here, the key difference, is that the Boeing 247 was the first airplane to be able to land based on its instruments alone. In other words, the conceptual shift was identifying the key piece of actionable and objective information to be able to get where you need to go. So with that in mind, let's start looking at the challenges we're facing in psychiatry and see how we can perhaps solve them. The first is that psychiatric disorders are currently made based on checklists of symptoms. Psychiatric diagnoses are currently made in this way. And so it's fair to ask, well, what does a psychiatric diagnosis explain? We looked at that recently by surveying quantitatively the world literature on brain imaging and psychiatric disorders, starting with brain structure. We looked at all the major psychiatric disorders and found that even though they differed widely in terms of their symptoms, they didn't differ so much with respect to the brain. In fact, what we found was a common signature, gray matter loss, in these 3 regions that was seen in all conditions. These 3 regions are not 3 random regions, but rather they form a network whose function it is to serve as a warning flag for the brain. Functions that warning flag are important for things like higher level cognition. So we next looked at patients in the scanner doing cognitive tasks. And again, across these multiple major psychiatric disorders, we found that there was very little difference in the brain. Rather, they had abnormal activation in exactly these same regions that gray matter was lost in. In other words, psychiatric diagnoses carry very little specificity with respect to biology. The same problem exists for our treatments. We use a one-size-fits-all approach. We assume that there's a drug for something like depression. Unfortunately, this too has started to break down. Irving Kirsch, 10 years ago, summarized all of the data that came into the FDA and found that the difference for depression between an antidepressant and a placebo was actually very, very small and below clinical significance. He then went on to write books exploding the antidepressant myth for us. But interestingly, this year there was another study doing the same thing in more people and came to the diametrically opposite conclusion, which is that, as you can see, their antidepressant drugs do work, and at the bottom it says, it's official, antidepressants are not snake oil or a conspiracy. Perhaps a sign of the times. But what this really illustrates is actually something far more interesting. The difference between drug and placebo in these two studies is identical. What has changed is our perception. Perhaps then these are all the wrong kind of questions to be answering or even asking. Maybe the question is not, does a drug work for depression, but can you actually use a treatment to define the patient? The concept is something like this. Within our clinical diagnoses, there's tremendous biological heterogeneity. If you had tools and objective measures to separate out that heterogeneity, something that's often been called personalized medicine, for example, based on a treatment, separate out sensitive type patients from resistant type patients, not only would you be able to guide care, but you'd be able to reveal mechanisms of the illness that are not evident from the clinical picture alone. Of course, from a practical perspective, these sensitive type individuals should go on and get that treatment that they were typed as being sensitive to. The resistant type individuals, well, it's not immediately clear what they should get, but we'll come back to them a little bit later. So let me give you some examples of this. In depression, we're part of a study called EMBARK. In this study, we've randomized a large group of depressed patients to receive Zoloft and antidepressants or placebo and image them at baseline to see if we can predict who will respond to one or the other. Collaborating with Madhukar Trivedi at UT Southwestern, we were able to find these sorts of signals. For example, as you can see in the blue line here, people who receive Zoloft have a decrease in their depressive symptoms over time that's very different from placebo. The resistant type people, however, show absolutely no difference in response to drug versus placebo. What was the function in the brain that mediated this? Well, it's the brain's ability to deal with these conflicting emotional stimuli, identifying the expression of the face while trying to ignore that word, a function that involved heavily a series of prefrontal regions. But this is true not just for drugs and in depression. This is true for post-traumatic stress disorder and PTSD. Here, the best treatment we have is actually psychotherapy. So we randomize people to receive either psychotherapy or be on a waiting list. And again found sensitive and resistant type people. The sensitive people in blue there responded very well to therapy compared to being on a wait list, whereas the resistant type people responded to therapy as if they were on a wait list. What was the function in the brain that mediated this? Very similarly, cognition, ability to code information into memory, and connectivity within that brain network that I showed you earlier was common across disorders. So if we think about our analogy, this is the beginning of the kind of objective tools, the kind of instruments that we need to start to be able to land our plane. Of course, this is only the first step. I think this will take us to maybe that Boeing 247 kind of conceptual shift, but are the kinds of things that I think are ready to start to scale and test outside the lab. But there are still even bigger problems we need to tackle. One of the big problems is actually that brains are not static. That is, that we've taken images of people's brains, single images of people, throughout the history of brain imaging and assumed that it stands in for the person writ large over time. And it turns out that's not entirely true. Russ Poldrak, who's a collaborating faculty member here at Stanford, when he was at UT Austin, he imaged himself twice a week for a year. And yeah, it takes a lot of dedication. What he was able to do is show that even trivial differences between, say, whether he had coffee and food before the scan or didn't, could be associated with differences in the connectivity of major brain networks. In fact, the problem gets deeper because it turns out that many of the measures we use in human neuroscience tend to generalize very poorly from the group level analyses, say, of depression as a group, to the level of individuals, which is the conclusion around which we want to draw to be able to conduct clinical care. That has led Russ, myself, and a number of other collaborating faculty members to develop out a concept that we call brain vitals. The idea is relatively simple, but not actually something that's being done right now. So if you take healthy individuals and define the variability in various important brain functions over time, you can define a healthy range of functioning. Now we know that For most neuropsychiatric disorders where this has been examined, when we look at clinical symptoms for diagnosis, we pick them up far later and after the brain signals that led to them, the brain abnormalities that contribute to the disorder. So in this toy example, you can see that you're following a person over time. When they start to leave the healthy range on a brain level, they can carry a brain diagnosis and only later be picked up clinically. But the really interesting thing comes when you take that signature for that individual, because now we're able to track at the level of individual people, and you can start running experiments on that person. We call that treatment. You can actually run randomized clinical trials within the individual as long as you have a signature that can respond fast enough, say a brain signature, much faster and more objectively than a clinical signature. In this toy example, you can see that maybe this person got several interventions in a blinded way, perhaps even, and it's the third one that worked, and maybe they relapse. You can test it again against that brain signature and treat them with that same intervention. The sort of motto, in many ways, of Silicon Valley is that if you can measure something, you can optimize on it. This is the goal here, is to be able to measure something so that we can optimize on it. There's a final challenge here. And this is the challenge of correlation versus causation. So it turns out this is not just something you debate in undergrad philosophy class. This has tremendous bearing on how we do human neuroscience, but also how we think about clinical care. So let me illustrate with an example. On this graph, is plotted from 1965 to 1980, the sightings of pairs of brooding storks versus millions of newborn babies born. Right? So obviously a very tight correlation, and from this it might be reasonable to assume that storks are how babies are made. Interestingly though, from this tight correlation, you can actually also equally assume that babies are how storks are made. So it's funny, it's cute. Actually, I think it is pretty cute. But the sad truth is that this is day-to-day reality in human neuroscience. What we do is we take pictures of the brain in action over time within individual, across people, and we try to build stronger and stronger correlations. We all know that big data are going to be a really important part of how we solve some of these problems. And there's been a rush in the field to get big data across many people, often in a very shallow way, but missing the fact that the goal of driving stronger and stronger correlations does not necessarily get you any closer to understanding cause and effect in the brain. It's a particular problem for human neuroscience. Neuroscience research in animals doesn't have that problem. Of course, that's not treating patients. So somewhere we have to bring these concepts together into a happy medium. In the end, I think a lot of what we've done in human neuroscience and in psychiatry is tell what I would call just so stories. That is, stories that are internally consistent but may not have any basis in fact. How do we get out of this loop? Well, one way to get out of this loop is to externally perturb brain activity, to push it around, to be able to understand cause and effect relationships between different brain regions, between the brain and behavior, and between the brain and symptoms. One of the ways that we do that in the lab is using transcranial magnetic stimulation, or TMS. TMS is a powerful electromagnet that pulses very briefly and stimulates the part of the brain that underlies that magnet. The intuition is as follows. If you stimulate part of the brain through its pattern of network connectivity, you can see the downstream effects that are the causal consequence of having stimulated that part of the brain. What you see here is a loop through just 100 milliseconds of brain time on EEG that follows a single TMS pulse to the prefrontal cortex. So we stimulate an image at the same time. It turns out, though, that TMS is not just a useful tool to understand the brain, it's also a useful tool to change the brain. If you give TMS in a repetitive fashion, you structure the pattern, you do that over days and then weeks, it can become an effective treatment. This has been developed as a treatment, for example, for depression. Since 2008, when this received FDA approval, there have been multiple machines produced now and sold on the market for treating patients with depression using this kind of intervention. But the interesting thing, and perhaps much like much of our treatments in psychiatry, this was developed without an understanding of mechanism. Again, the concept is if you can measure something objectively, you can now start to optimize on it. So we decided to do that recently by combining this probing of circuitry, single TMS pulses with brainwave or EEG recordings, before and after patients got this treatment. What you can see on the bottom there is the brainwaves that are induced after a single TMS pulse, which begin at just 30 milliseconds after that pulse. If you compare the response of patients before and after treatment, what you see is that even that earliest response, 30 milliseconds after the pulse, is suppressed by real but not sham TMS. But the most exciting thing is that the change in EEG tracks clinical change. Again, if you can measure it, you can optimize on it. It's giving us great hope that we can actually individualize and personalize this treatment for depression, but that's really just where the fun starts. You can then take this tool and go elsewhere in the brain. One of the challenges in neuroscience in humans has been that much of the brain is so-called non-eloquent. It doesn't tell you what it does when you disrupt it. You need to be able to read out brain function, for example, with simultaneous imaging. Here, we're stimulating different parts of the brain and reading out those cause and effect relationships, creating what I call a causal connectome of the brain. That's a useful basic science tool, but it's also a useful clinical tool. You can discover things that are not evident just by doing the imaging alone. For example, in patients with PTSD compared to healthy individuals, we compared response to stimulation at various parts of the brain. We found that if you stimulate the right prefrontal cortex towards the front, the neural response you get, here in blue in patients and in red in healthy individuals, differs. In fact, patients here have a blunted response in a particular way that already suggests mechanisms that underlie this. You can then start looking around the brain and say, how important is it that you have to be right on that target? What happens, for example, if you stimulate another region in the right prefrontal cortex? Turns out, if you do that, there's no difference between groups. What about going to the left hemisphere, stimulating the same region in the front of the left prefrontal cortex? And the answer there is, again, no difference. So doing this kind of thing has revealed a very exciting new target, both in terms of the brain and in terms of the brain's response, around which we can develop new interventions using this tool. So let me pull this together for you and kind of take stock of where we're at and where we're going. In order to advance what we'd call precision psychiatry or personalized medicine for psychiatry, I think we can start drawing on some really exciting examples here. For example, the case I gave you around depression and Zoloft prediction, or around PTSD and psychotherapy prediction, to be able to create those first and very basic instruments, that 247 model of just those first things that allow you to get to where you're going in an objective way. This, I think, is ready to scale and really ready to be tried in the world outside the lab. But this is still the 247. We want to be able to get to that 787. We want to be able to get to the full sophistication that the plane can go on autopilot, and you start to wonder, is there even a pilot there? Is this thing going completely on its own? To do that, we have to ask the question of, how do we actually transform psychiatry? How do we rethink our basic questions? Taking inspiration from Michael, how do we actually ask the questions that need to be asked and see where some obvious alignments could happen? To get there, I think we have to think about especially those people who don't respond to existing treatments. And we know there's a lot of those. And really get to the point of identifying circuit abnormalities in individual people, using tools, for example, like the Brain Vitals or these causal imaging tools that I told you about. The purpose here is to be able to link much more closely brain signatures and brain interventions so that at the individual level, we're able to create circuit targeting treatments, including for the individual, that seek to improve their symptoms, improve their behavior, and solve the challenges that they've been dealing with often for years. What this requires then is a whole new phase of tool building. If we can start to identify those questions, now it becomes an exciting new endeavor to build those tools to see this promise happen. Hopefully this gives you a more hopeful perspective on this vexing and big challenge of mental illness. And I look forward to discussing it further with you in person afterwards. Thank you.